Hello, and welcome to Cell and Gene Live. Today's topic is the therapeutic potential of exosomes. I'm Erin Harris, Chief Editor of Cell and Gene, and your moderator for this event. Your time is well spent here with us because our panel of experts is just incredible. Joining me today are Dr. Mari Mitrani. She is the co-founder and Chief Scientific Officer of Organicell. Dr. Benny Sorensen, he is the Scientific Advisory Board member of Codiak BioScience, and Nicolas Rousseau, co-founder and CCO at EVerZom. So we're going to get started in just a second, but before we do, I have a few quick housekeeping items to go through with you. We absolutely love to hear from our audience, so please utilize the Q&A function in the box at the right of your screen to submit questions. So don't be shy. I will get to as many as I can throughout our session today. Watch out for our poll questions. They're going to pop up on your screen during the session to add a little bit of depth to our conversation today. So look out for those. If you have a specific challenge or question that isn't addressed on today's session, feel free to reach out to me directly via the link below my headshot on the left of your screen. Please take advantage of the resources we've curated for you. You'll find those in the lower right side of your screen. And lastly, if you have any technical difficulties, click on the question mark icon at the center bottom of your screen for help, and we will get right to it. All right, so without further ado, let's get going. First, I want to push out that first poll question. So go ahead and review that, and I will get to those responses in just a bit. But we're going to head over to our discussion now. So, all right, as experts in the field, I want to talk to our panelists about level setting the sector's understanding of extracellular vesicles right from the get-go. So, Benny, I'd like to start with you. Can you give us a little bit of background on exosomes and bring us up to where we are today? Hi, Benny, are you there?
Can you hear me, Erin?
Now I can, yes.
Okay, well, I think it's a great first question, and I think the very short answer is that we have really made a leapfrog. We've really moved this field from, thinking that extracellular vesicles was just kind of waste disposals to really believing strongly that they are nature's drug delivery vehicles. And I think over the recent 5 years in particular, we've further kind of got a deeper understanding of the biological role of exosomes, in particularly in overall immune surveillance, meaning that, a key role of the function of exosomes is really to communicate with and orchestrate immune system kind of responses. At Codiak BioSciences, that's a biotech company with a very dedicated vision and mission of transforming the biology of exosomes into a new category of biologics medicines. And I think key discoveries that we have made over recent years are really unique exosome proteins such as PTGFRN and BAS1. These are proteins that has enabled building a highly versatile engineering platform that's called NGEX. And it is really to in a very modular fashion, design exosomes with bespoke pharmacological properties, adding multiple different type of drug moieties on the inside or outside of exosomes, such as proteins, peptides, antibodies, small molecules, as well as oligonucleotides. So I believe that we are kind of standing at a, if you will, key inflection point in the journey of transforming exosome biology into medicines. I think Codiak has been leading this by starting 2 clinical trials with engineered exosome therapeutics within the last year. And I think as we will discuss today in much more detail, really the therapeutic opportunities are multiple. And I expect that we'll see a wide breadth of exosome therapeutic entering the clinic over the next month and years to come. So very, very exciting points and very exciting inflection we are standing at.
For sure, for sure. So the title of this webinar is The Therapeutic Potential of Exosomes. So we want to cover what is their therapeutic potential, right? So Mari, I'd like to start with you. Can you kick us off here? Explain to us in detail the advantages of exosomes and what in fact is their therapeutic potential.
Thank you so much, Erin. And as Benny was saying, I think it's very, very important for us to understand that these extracellular vesicles are these nanocytes, little compartments that contain a specific message. And that is what me and my team have been doing for the past several years on understanding what is the cargo and how, if we understand the cargo, we can impact patients' lives. So me and my team have been really characterizing what's inside of them, and most specifically inside of that microRNA, what are the pathways that can actually lead into therapeutic responses. We are really honing down after all of our preclinical data, our animal models, and published studies that the base of anti-inflammatory compositions that these microRNA channels have, it's remarkable. And unfortunately or fortunately, last year with the pandemic, we were able to transition and pivot from all of our lung injury model platform into we were studying neonatal damage, and we were able to transition into the COVID arena and understand how we can actually impact anti-inflammatory and immunomodulatory, as Benny was saying. These exosome cargo and the information that's inside of these exosomes can actually help us in a bigger scheme of a therapeutic component.
Nice, nice. Okay. And Nicolas, I want to make sure we give you the opportunity as well to comment here.
Thank you.
From your perspective, yeah, go ahead.
Yeah, well, I think maybe first as the therapeutic potential, we could say to complete on what Organicell and Codiak says. What is very amazing with those exosomes is that they are a broad therapeutic platform, and you can use them in many different pathologies in regenerative medicines. Either lung regeneration, immunomodulation, but some companies are working on osteoarthritis, fistula healing, burns, ulcer, diabetics. So just for me, the potential is totally huge. So you have this first potential in regenerative medicine, in which you are just using naive exosomes, but it is all the more impressive how you can pimp up your vesicle, try to load it, try to surface engineer it for drug delivery. So it's quite, I would say a very powerful platform that needs to be really well adapted, and we still have many things, I think, to discover how to fully use this platform.
Sure, sure. And given that there is still much to be done, I guess this is a bit of a forward-looking statement, but how can or will exosomes change the way patients are treated with various diseases? And Benny, if you could kick us off with your thoughts on that.
Yeah, I think, Erin, and I hear everybody on the panel kind of also clearly stating this. I think in brief, the potential of an exosome therapeutic is really to drug what has previously been undruggable, and achieving that by customizing really selective delivery, targeting specific cells, loading and or kind of cloaking, if you will, potent and very complex pharmacological payloads, and then leveraging that, exosomes are immune silent, and that allows for repeat dosing, and you can further then leverage that exosomes kind of uniquely have an ability to stay within select kind of biological compartments, and thereby and actually they're really leverage that you can target and deliver unique payloads. And I think, in other words, what really is going to benefit patient here is this ability of now forward engineering or actually hijack nature's natural drug delivery kind of vehicle, or natural, if you will, popular speaking, FedEx system to really, enhance the therapeutic index. And enhancing therapeutic index kind of loops back to where I started, will now finally enable us to drug what I think has previously been kind of undruggable.
Mm-hmm. Sure. Before we check out the results of the first poll, Mari and Nicolas, I want to make sure I give you the opportunity to comment there as well.
Yeah, I think, as Benny was saying, as being in the cell industry for so long, would understand that the scalability and being able to reproduce a drug is very difficult. It's still very difficult and will maintain to be very difficult. But when we transition into the exosome arena, it is, and we are proven that it is a way easier to scale and mass produce a product that can have the same reproducibility in terms of a final product or a final drug. And that is what we're proven, and this year has proven that we can do so, right? We're conducting 5 different IND trials and being able to deliver at the bedside in a way that is not as complicated as cell therapy was, where you have to have these chemicals, DSMO, that you have to wash, that you have to make sure that you clean those, that our cells will going to be viable once you thaw them out. Exosomes do not have that problem to face, and I think this is what it makes us ahead of the curve in this regenerative medicine arena.
Sure.
Yeah,
that makes sense.
And maybe to complete on that, what I think is the main difference with cell therapy, as Dr. Mitrani has said, is that they are not living cells. They are not living entities, so you can easily freeze them. Some team are also freeze drying them. So you are much more closer than a traditional pharmaceutical product than difficult to handle cell therapy. You can also couple them with, in the field of regenerative medicines, you can couple them with biomaterials that will help them to be easily delivered. So either biomaterials, hydrogel, or matrix, and I think it unlocks the potential of stem cells really for the regenerative medicine part.